Okay, we'll give it a couple more minutes, but while we do that, I'll just run through some housekeeping while we wait. So yeah, I can see the numbers are still climbing here. So let me just say that today's session is being recorded and an on-demand version of the webinar will be provided shortly after the webinar concludes. All the microphones have been muted, apart from mine and Josh's, but we have a chat box available on the right-hand side for you to submit questions to as we present. There's also going to be some polls going on the right-hand side there. So please check those out and we'd love you to respond to them. If you are viewing the webinar in a windowed mode as well, yeah, please take a moment in the top right hand of the screen. You can just move your mouse there and you'll be able to put the whole presentation into presentation mode and you'll be able to see it full screen. Also on the right-hand side, we've got of the screen, we've got a library, a resource library with some useful resources for you to access quickly. We've got many more tools and resources available on metabolon.com, particularly in the applications and the learn menus. So if you're interested, please go and take a look there as well. We will have a short Q&A session at the end, depending on how much time we've got. So please use that Q&A tab over on the right-hand side to submit those questions. Please note, though, the questions are moderated and we will respond to everyone. If we run out of time, though, to get to all of the questions live, we will follow up with you directly after the webinar via email. Okay. We give it 30 more seconds and then we'll get going with the presentation. Okay, so without further ado, it's my great pleasure to present to you the latest new product announcement from Metabolon, the microbiome research solution. My name's Joe, and with my colleague Josh, we'll be guiding you through the importance of microbiome research and the insights available when we combine gold standard metagenomics with industry-leading metabolomics for a greater than the sum of its parts result. On the webinar, we're going to cover a few topics. We're going to look at a quick state of the union on microbiome research to understand its importance and its aims. We're going to look at the combination of metagenomics and metabolomics with some case studies. We'll take a look at an overview of Metabolon's new microbiome research solution, and then we're going to go into a deep dive into the microbiome analysis tool, which is a new tool within the Integrated Bioinformatics Platform that some of you may have seen in previous webinars. Finally, there'll be a short summary before we move on to any questions. So as a reminder, please feel free to ask questions in the Q&A on the right. We'll be collecting them throughout the webinar, and also don't forget those polls as well. Right, let's get started. So explosive publication growth, the first regulatory approvals, deep therapeutic pipeline, and a really rapidly expanding diagnostics market all show that decoding the microbiome is a really core driver of modern biomedicine. We've seen publications in the microbiome space up 5x in the last 10 years. Today, we've got 2 FDA-approved live biotherapeutics. There's a really healthy pipeline of drug candidates spread over not just a couple of big players, but many, many companies. The microbiome diagnostics market is also set to grow 3x over the next 8 years. And we've seen a tremendous increase in direct-to-consumer offerings that focus on microbiome. And the list really goes on with patents and funding growth as well, much of which has been really driven by huge reductions in sequencing costs that make it more viable than ever to interrogate microbiomes at scale. When it comes to interrogating microbiomes at scale, it's really next-generation sequencing, specifically 2 methods, which have dominated the rich landscape of microbiome discoveries: 16S amplicon sequencing and also shotgun metagenomic sequencing. So in 16S targeted sequencing of the hypervariable regions of the 16S rRNA, it's performed using amplicons to produce short reads. These variable regions really differentiate bacterial and archaeal taxa to produce a profile of what microbes are present in a sample and their relative abundance. 16S has really proven to be a cost-effective approach capable of interrogating the microbiomes of sample types regardless of interfering host genomic DNA. Shotgun sequencing, on the other hand, really randomly captures sequencing fragments across everything in the sample, including that host genome. So delivering typically higher resolution in identifications at species and strain level compared to the genus level. This technique is ideal for linking composition to functional potential as well. By virtue of sequencing microbial genomes as well, that functional potential can be directly identified. This technique of shotgun really comes at a higher cost than 16S and introduces some complexity in selecting sequencing depth when working with different sample types, particularly sample types with a high host genome content that essentially dilutes valuable microbial reads out. NGS-focused microbiome studies typically aim to answer really these three questions. Which microbes are present, often depicted as a cladogram shown here, gaining taxonomic resolution as we go further towards the edge? How abundant are they? And that's illustrated with this relative abundance bar chart here that compares, in this case, two different sample groups. And also, what can they do? Understood by either interrogating the identified microbes' reference genomes in public databases, or in the case of shotgun sequencing, really directly identifying genes and their predicted or known functions. Really, though, what we need to focus on is that the genome of an organism can do what it actually does in, what we need to focus on here is that the genome of an organism really defines what it can do, but it leaves a lot of room for guesswork of what's actually happening in a sample. So that's where we enter metabolomics and the concept of microbes as a mini metabolite producing machine. If we zoom out for a moment and take a look at the big picture, it's really the genome, the transcriptome, the proteome, and the sum of the environmental and lifestyle effects that are vital parts of the complete biological system. All of them ultimately impact the metabolome, which is the definitive representation of the phenotype. The microbiome in particular plays a crucial role and is a key driver of the metabolome, in that microbial metabolites are key mediators of microbiome host interactions, influencing things like health, disease, treatment response, and overall well-being as well. As I'm sure you can all appreciate, microbes and metabolites are inextricably linked. And so when their profiles are combined, they unlock the potential to really deeply understand biological systems. While metagenomics determines that functional potential of a microbiome, it's metabolomics which reveals what's actually going on. To exemplify this, I'd like to briefly touch on a case study by a collaborative group, mostly out of China and Sweden, who looked at the microbiome metabolome dynamics associated with impaired glucose control and responses to lifestyle changes. We know that type 2 diabetes is a heterogeneous disease, shaped both by genetic and environmental factors, including the gut microbiome. But a mechanistic understanding of these microbial drivers of the disease remains elusive. To better understand the microbial drivers of type 2 diabetes, this group used fasting blood glucose or oral glucose tolerance screens to screen both individuals in a discovery, but also a validation cohort as well, and evaluated the predictive power of three main factors, including 193 dietary variables. There were 34 clinical factors, and also 1,427 metagenomic species on each of the 978 metabolites identified in that discovery cohort. They ultimately identified 502 metabolites that were linked to altered glucose control, 143 of which correlated to the gut microbiome. And a subset of these metabolites were able to accurately identify both prediabetes and type 2 diabetes, more so than just the metagenomics data alone. Overall, this study shows how metabolomics is integral in connecting those mechanistic dots between diet, microbial composition, and glucose tolerance. The discovery cohort used here was from the Impaired Glucose Tolerance Project, and the validation cohort was the Swedish Cardiopulmonary Bioimage Study. Highly recommend this paper to you all. Take a screenshot now if you'd like to know what that is and have a read. Metabolomics and metagenomics also go hand in hand in that clinical trial setting, as seen in this phase 1b study as well. It was evaluating SIR287 from Seres Therapeutics. In the Human Microbiome Project, Firmicutes bacteria were found to be particularly abundant in healthy adults. And these bacteria produce metabolites that really help maintain that intestinal barrier. Ulcerative colitis patients have a lower microbial diversity, including much lower abundances of Firmicutes bacteria. Based on this, Seres Therapeutics developed SIR287, which is a drug containing Firmicutes spores, and found that this drug was both effective and safe compared to a placebo group. They identified microbe-associated metabolites with relevance to gut homeostasis, to mucin production, to epithelial barrier function, and immune signaling, including metabolites such as hexanoate, taurine, and arachidonate as well. The metabolite profiles were distinguishable when looking at pre and post SIR287 treatment, as well as in remitter versus non-remitter sample groups. Overall, they indicated that the constellation of metabolite associations with SIR287 and clinical remission aligned with emerging data from several other ulcerative colitis cohort studies, and that these metabolites are likely contributors to SIR287's mechanism of action, which they aim to explore further in a larger phase 2 study as well. So from those exciting results from my collaborators, let's move on now to look at Metabolon's newly launched microbiome research solution. So continuing our theme this year of integrative multi-omics, the new microbiome research solution combines metabolomic and metagenomic data generation with bioinformatics and interpretation reports for a really end-to-end experience. For metabolomics, a dedicated panel optimized for cost-effective profiling of key metabolites has really been created as an alternative to our flagship untargeted global discovery panel. We've also added metagenomic sequencing services right-sized to your research goals, including 16S rRNA sequencing and shotgun metagenomics at a variety of different depths. To marry these two data modalities together, there is a significant update to the Integrated Bioinformatics Platform to include metagenomic sequencing QC, data processing, identification, and also that critical integrative multi-omics analysis as well. Finally, in the microbiome research solution, we've extended our repertoire of tailored reports written by our scientific experts to include curated multi-omic results that inform key hypotheses identified during that study design phase. Together, these 4 services really create an end-to-end offering for the discovery of deep insights into host microbiome interactions. Let's briefly dive into a little bit more detail on that microbiome focus panel created as an alternative to the global discovery panel. The microbiome focus panel measures approximately 450 metabolites with relative quantification, and these metabolites were selected for microbiome activity in either fecal or blood samples. They cover a wide range of key pathways implicated in microbiome research, such as secondary bile acid metabolism, tryptophan metabolism, and a fully quantitative panel of 9 short-chain fatty acids, just to name a few of the elements of coverage here. Obviously, though, not all metabolites are expected in every sample, and from the wealth of Metabolon's historical data, we expect to see about 225 metabolites in a plasma sample and 275 in a fecal sample. So on top of the additions to our metabolomics offering, metagenomic sequencing can be included as part of the microbiome research solution as well. You can choose from 16S amplicon-based sequencing or shotgun metagenomics at 4 different depths. We've got shallow, balanced, deep, and ultra deep, offering progressively higher read depth in each level here. There's also an option to bring your own shotgun or 16S data. This is done via a simple upload that will ingest it into the Integrated Bioinformatics Platform, ready for analysis with your metabolomic data from either the Global Discovery Panel or the new Microbiome Focused Panel. And for those that haven't worked with Metabolon before, I'll say that projects at Metabolon have a turnaround time of only about 12 weeks for data that's ready to interpret and available inside of that Integrated Bioinformatics Platform. Additional advanced multi-omic interpretation reports are available within 4 weeks post-delivery as well. So talking of the Integrated Bioinformatics Platform, or IBP, you might hear us call it for short, we've developed an entirely new microbiome analysis tool, providing gold standard microbiome analytics from raw data all the way to that integrated multi-omics piece. The microbiome analysis tool is released as what we call a beta with free access for any projects booked during the beta period. There's already a demo data set, so if you've not already registered for an account at My Metabolon, head over to metabolon.com as soon as I finish speaking or after this webinar and get exploring the demo data set with all of the functionality that we're about to see. So in terms of that functionality, the microbiome analysis tool covers all of the essential questions we typically ask of a microbiome data set, along with tools to integrate metabolomics for a deeper look into mechanistic insights that differentiate your sample groups. Questions like, how does the microbiome composition vary within and between my groups? How does abundance change as we view more specific identifications? What bacteria are present in what abundances? Which bacteria abundances change the most between different groups? Which microbes and metabolites are significant and are they correlated? And which microbes share metabolomic signatures and vice versa as well? So lots of utility there. With that, it's my privilege to introduce our senior microbiome scientist, Josh Simpson, the principal creator of the microbiome analysis tool, for a demonstration of its functionality. Give me a moment here to stop sharing my screen. And okay, take it away, Josh.
Thanks, Joe. So, today we'll navigate through the Microbiome Tool interface within Metabolon's Integrated Bioinformatics Platform, or IBP. I'll be guiding you through each screen of the Microbiome app, and by the end, you should have a clearer understanding of the full suite of 12 metagenomics-focused tools available and how integrating metagenomics data with metabolomics data can reveal deeper biological insights. So right now on my screen, you're looking at the dashboard of the microbiome application within this IBP. This is a cloud-based, code-free platform that integrates the best-in-class analytical tools for the microbiome and is designed specifically for streamlined multi-omics microbiome research. Our goal from the start has been to combine intuitive usability with rigorous scientific standards. And as Joe mentioned earlier, our platform provides flexibility for where you decide to get your metagenomics data. You can submit your samples directly to us for complete metagenomic processing, or you can simply upload your raw metagenomics data to our online portal. Our workflow then automatically processing the sequencing data to generate your bacterial community profiles. These data are then harmonized with their corresponding metabolomics data sets. And after that, the workspace we provide you to explore the data is entirely point and click. So even if bioinformatics isn't your primary expertise, you can confidently conduct advanced analyses, which we hope empowers a broader scientific community to engage in microbiome research. On the navigation menu here, you'll notice dedicated sections for quality control and detailed analytical tools. I'll be exploring each of these sections as we proceed. And the bottom line here is that we're your one-stop solution, handling all data processing complexity so that you can focus purely on scientific discovery. Now, let's discuss the essential first step of any robust analysis, data quality. This is the metagenomics quality control section of our microbiome app, where we've used FastQC and MultiQC to generate this interactive QC report that summarizes the critical quality metrics of your sequencing data. For those in attendance who have experience with metagenomics datasets, these assessments should be quite familiar, but here we provide them in an interactive and exportable format for convenient review. And I'd like to note that if any of your samples here appear as outliers, our team of scientists are available to collaborate with you on determining the best path forward for your downstream analysis. Now that we've confirmed the quality control of our samples, we're able to launch an analysis of our metagenomics and metabolomic samples. And from here you can define your analysis based on any metadata trait for your sample set, including metadata columns like diagnosis, define which group are compared, define which taxonomic rank you're wanting to analyze, as well as alter prevalence filters and specific statistical methods that are used to define the way that your samples are analyzed. Then you just decide on an analysis name and click submit, and the analysis is ready to launch. So we've generated an analysis comparing IBD patients to non-IBD patients at the species level. Now let's take a closer look at their bacterial community profiles using the alpha diversity tool. This screen presents a box plot comparing an alpha diversity index with the y-axis representing the diversity index itself. So if the box plots are at different heights, it suggests a meaningful difference in community structure. There's also an option to switch between different diversity indices using this drop-down menu on the right. And here I've selected the Gini index, which measures equality of a distribution of community, with a value closer to 0 indicating a better balanced community. So as we might expect, the non-IBD group on average has a much lower Gini index compared to the IBD group. We can also further subset our data according to different metadata characteristics, such as sex, to explore homogeneity of a group within our cohort. And these results, along with results shown for any of the figures in any of our screens, can be exported as an Excel file, a CSV file, or any of your other favorite data formats. Finally, we offer the ability to separate individual data points from their overall box and whisker or violin chart to further explore how individual donors contribute to the overall makeup of your groups. Now, let's explore beta diversity. This is beta diversity, which assesses the differences in microbiome community composition between samples and groups. On the screen, we show beta diversity in an ordination plot using principal coordinates analysis. So each point represents a sample, and the distance between the points reflects how dissimilar those samples' microbiomes are. Each axis is a different principal coordinate, and those can be modified in the column on the right. In our example, purple points, the IBD samples, cluster towards the left, and several of the green samples, the healthy donors, cluster towards the right. This separation indicates that the overall microbiome composition differs between some members of the two groups. We've enabled the option to show a confidence interval ellipse for users to get a better sense of the extent of the overlap between their cohort groups. Also, a simple toggle switch can highlight outliers across your ordination plot. And for additional customizability, users can modify the dot size and other aesthetic controls as they wish. These and all the other plots can then be exported at the click of a button. Now, let's explore those diagnosis differences a bit further using a cladogram. So we know from beta diversity that communities differ, but where in the taxonomic hierarchy do those differences occur? This cladogram is a circular dendrogram of the microbial taxa within our sample group. The size of each circle represents the abundance of that taxa, and we can use this representation to explore the abundances of specific taxonomic ranks within the groups of our cohort. In this example of the IBD group, we can clearly see that at the phylum level, the abundance of firmicutes in green is markedly less than that of the Bacteroidetes in yellow. These differences become even more apparent when we customize the plot to color bacteria beginning at the class rank. These results suggest that the Bacteroides in the IBD group are the primary contributor to the low Gini index that we observed back in alpha diversity. To better identify the most abundant of these Bacteroides, we can use our minimum abundance filter to modify our cladogram to only show taxa exceeding our specified abundance threshold, by again using the circles as our guide, we can easily and efficiently identify the specific genera and species that are driving the dominance of Bacteroides in the IBD group. Now, let's take a look at the relative bacterial abundance screen. While cladograms show differences in the tree view, relative abundance stacked bar charts provide a more nuanced look at the data across the whole cohort. So each color represents a unique taxa, and each bar represents a unique sample. I've initially sorted these by diagnosis. However, removing group separation causes the samples to be reorganized based on similarity of microbial profile alone. This enables us to explore whether certain study variables, like diagnosis, contributes to distinct sample clustering. These variables, like diagnosis, can be viewed according to the colors on the bottom. In this case, we again see that many IBD individuals have Bacteroides dominant microbial profiles, and we also see that a subset of non-IBD individuals are lacking the taxa altogether. These are likely the non-IBD samples that diverge from the IBD samples the most dramatically in that beta diversity screen earlier. We can easily select another metadata characteristic on the right to explore whether the differences we're observing extend to other study variables. For instance, grouping the samples by sex here results in relatively comparable distribution between the groups, which suggests that the observed differences in the IBD diagnosis groups are driven more by diagnosis than by sex. Finally, it's worth highlighting that this screen enables users to efficiently explore low abundance microbes by zooming into the plots with a single click. And hovering over individual taxa in this zoomed-in area provides precise abundance values for each sample, which offers an even more granular view of the data. Next, we'll check out the Hierarchical Clustering tab. Hierarchical Clustering Analysis, or HCA, is an unsupervised method for grouping samples based on the similarity of their taxonomic profiles. It organizes samples into this tree-like structure shown on the left and the top, which is a dendrogram. Samples with similar microbial compositions are positioned adjacent or nearby to one another. This approach reveals natural groupings within your datasets and highlights clusters without relying on predefined categories such as metadata characteristics. On this screen specifically, the heat map represents the relative abundances for the top microbes across your cohort at the species level. The dendrograms and heat maps together provide detailed insights into sample similarity and the relative abundance of individual taxa across samples. Based on the study variables indicated in pink and blue on the y-axis of the left of the plot, we can begin to identify areas where the samples cluster together and areas where samples diverge the most in terms of taxonomic composition. As with the other screens, we have several parameters we can use to reformat the information shown in the plot based on our observations across the other screens. For a closer look at the most abundant taxa across the cohort, I can reduce the plot to only show the top 5 most abundant species, and doing so reveals a subset of members of my cohort that have markedly low abundance of these species. Likewise, the majority of the cohort has a lot of these species, as would be expected. Generally speaking, we can use this plot to naturally group samples based on microbial profile similarity. This highlights the taxa that are consistently different in abundance between conditions or phenotypes, despite being among the most abundant taxa across the cohort overall. Now let's explore the ANCOM volcano plot, which enables you to determine which specific taxa are significantly different between groups. ANCOM, or Analysis of Composition of Microbiomes, is a robust statistical test designed to identify microbial taxa that differ significantly in abundance between two groups. Is particularly well suited for microbiome data because it accounts for compositional nature of these data sets, which reduces false positives and provides a clearer picture of meaningful differences. This plot is structured like your typical volcano plot, with each point representing a microbial taxon, in this case species, and the x-axis showing the log 2-fold change in abundance between the two groups that we specified when we launched the analysis. So IBD versus non-IBD in this case. And we capture how much a taxon's abundance shifts between the groups. So the y-axis represents the W statistic, which is a measure of statistical support that indicates how consistently that taxon differs across many pairwise group comparisons. As with the other screens, we offer users the ability to zoom in and highlight specific points, each representing specific taxa, to be able to deeply explore which individual taxa significantly vary between their groups. These plots can be further customized by the options shown on the right, and as with the other plots, the figures can be exported and saved. After identifying the specific taxa that significantly differ between your study groups, you can explore these results in the context of broader biology using our taxon set enrichment analysis. This is a powerful tool for connecting taxonomic shifts to their broader biological roles and functional impacts. It works much like gene set enrichment analysis in genomics, but instead of genes, we're looking at groups of taxa with shared biological characteristics. This approach provides a higher level perspective, helping us move beyond individual microbes to understand how groups of functionally related organisms have the potential to change together. Unlike the Ancon volcano plot, which highlights individual taxa that differ significantly between the groups, this tool identifies collections of taxa that collectively exhibit meaningful shifts across your cohort, which offers a deeper and different biological context. In the case of our current dataset, you can see that one of the top associations is with the set of microbes that are elevated in Crohn's disease relative to healthy individuals, which makes sense given the context of our IBD study. As you see, these results can reveal broader functional themes in the data, which moves us from taxonomic changes to metabolic and physiological interpretations. We offer multiple databases for this analysis, including GMRepo v2 and BugSigDB, and as you can see, we also offer a wide range of customization options, including color, as well as the ability to change the visualization to a bar chart from the circled plot I showed a moment ago. Those were our metagenomic analyses, so now let's integrate that data with metabolomics data to explore the functional consequences of those microbial shifts. So Diablo is a supervised multi-omics integration method that identifies the biological features, in our case the microbial taxa and the metabolites, that are co-associated with different study groups, and it can be used to uncover the combinations of variables that actually best separate the groups, in our case, IBD versus non-IBD. In other words, Diablo helps us develop models that link microbes and metabolites to our study groups, which can then help highlight the key drivers of host phenotypes. So here we show the Diablo scores overview, which is a similar view to what was shown with beta diversity. Each point is still an individual sample, however the factors underlying each graph are now comprised of both metagenomic and metabolomic features. So each component on the x and y axis represents a unique grouping of these features, so the components that result in the strongest divergence of our groups are inherently driven by the features in our dataset that best separate our groups. We can expand the scores overview plot to view more components at the same time, which for our dataset highlights that components 1 and 2 seem to have the greatest divergence between groups. Let's take a closer look at those components in the Sample Plots tab. In our Sample Plots tab, we can get a closer view of the components from the previous screen, but with more customization options that I hope become familiar from the rest of the app. Here we can also view group separation driven by 3 components in 3 dimensions. As with the other screens, hovering over any single data point displays the sample name for that data point, which can then be used to track specific samples between the screens within the app. Now that we've explored components 1 and 2, let's examine which features substantially contribute to these components. We do that using the Feature Importance tab. Here, we can see that Bacteroides uniformis, among other Bacteroides, are the main features driving separation between groups for component 1. We can also see on the right that this feature is elevated in the IBD group and likewise depleted in the non-IBD group. In this example data set, the metagenomic features seem to contribute more heavily to component 1. However, we can still view the metabolomic features that contribute to component 1 by selecting metabolomics as the omics type in the toggle on the right. If we switch to our loadings plot, we can get a more quantitative view of how these individual features in our feature importance plot quantitatively contribute to the component. After that, we can use the Model Performance tab here to explore the ability of our components of interest to accurately differentiate between groups within our cohort. Finally, let's further explore the associations driving group separation using the Circos plot view. This visualization highlights your associations between microbial and metabolic features as arcs, connecting them in a circular layout. Each node around the circle represents a microbe or a metabolite. If Diablo detects a strong link between a microbe and a metabolite, a line connects them, with here the orange lines indicating positive correlations and blue lines indicating negative correlations. If you're interested in only viewing the strongest correlations, we can use a slider on the right to define our correlation threshold, and we can also explicitly filter our plot to only show the top N associations. Here, doing so highlights that Bacteroides ovatus is negatively associated with the commensal E. ramosum species. This suggests that these may be competing taxa in the gut, and that the ability of Bacteroides to diminish the presence of E. ramosum could contribute to its dominance in IBD populations. The discovery of this relationship can also serve as a primer for you to generate new hypotheses for downstream experiments and/or mechanistic validation. Our final set of integrative analysis screens are comprised of correlation integrative analyses. These explore the strengths of correlations, either Spearman or Pearson, between all individual pairings of microbial taxa and metabolite abundances across the cohort. The correlation values quantify how strongly each metabolite varies as a function of each microbial taxa abundance across the dataset. These analyses help uncover microbe-metabolite relationships that are consistent across the full cohort, suggesting persistent biological mechanisms and offering support for key associations observed in metagenomics or metabolomics alone. So what I'm showing here is a hierarchical clustering heat map, which clusters similarly to our earlier metagenomics clustering screen. However, where that one had samples, we have metabolites. So every point on the heat map represents a correlation coefficient between a microbe and a metabolite, and these metagenomic and metabolomic features are clustered based on their similarity of their correlation profiles. We also give you the option to display relevant contextual information for each one, such as superpathway labels for metabolites or higher-level taxonomy for microbes. This approach helps you identify natural groupings of co-regulated metabolites and microbes, which can highlight metabolomic activity that occurs as a function of taxonomic rank. Our cluster correlation heat map goes a step further with this information by grouping microbe-metabolite pairs into discrete clusters of features based on the similarity of their correlation profiles using the spectral biclustering algorithm, which divides the heat map into discrete grid spaces based on the clustering of the features within. As with the last screen, you can choose to view this based upon Spearman or Pearson correlation coefficients, and the results shown in the plot can be filtered further based on your preferred statistical thresholds or how many groups you actually wish to show on the plot. So each number on the plot represents a discrete set of microbes and metabolites that are co-associated across your cohort, and each group can be examined further using the bicluster member table that's beneath the plot and available for export. Our final integrative clustering screen is the embedding screen, which provides a complementary view to what is visualized in the other metagenomic metabolomic correlation clustering heat maps. So while the integrative heat maps directly cluster this metagenomic and metabolomic data set based on similarity correlation profiles, the embedding screen uses dimensionality reduction such as, and including, PCA or UMAP to project feature correlations into a lower dimensional space. Clustering algorithms are then applied to group similar data points. Here we provide the options of k-means or dbScan as clustering algorithms. And the resulting cluster assignments are used to color individual features on the plot. Embedding highlights microbe-metabolite connections that may not be evident from correlations between individual metabolites and individual microbial taxa alone, and it can reveal additional coordinated patterns within the datasets that might not be obvious to us upon initial looks. It can also reinforce and complement key associations observed independently in your metagenomics or metabolomics datasets. With these clustering tools, along with the full suite of analytical screens in our platform, our goal is to empower you to explore your microbiome datasets efficiently and comprehensively. Our metagenomics tools quantify who is there by measuring the abundance of genes attributed to distinct microbial species, while our metabolomics analyses reveal what's happening by capturing the molecular phenotypes at the microbe-host interface. And together, these approaches synergistically reveal how and potentially why these phenotypes arise. insights which are often invisible when analyzing each data type in isolation. Our platform consolidates these data sets, your metabolomics, your metagenomics, and your sample metadata, into a single code-free environment that provides intuitive point-and-click analyses without the need for extensive data wrangling or engineering. Throughout this workflow, you have access to Metabolon's industry-leading metabolomics data quality and bioinformatics software, which ensures that everything you find in your data is built on a foundation of rigorous high-quality data. From raw sequencing data to visualization and interpretation, our platform handles every step without manual intervention or coding on your end, while still providing you with the flexibility to customize analyses to suit your specific research questions and statistical preferences. Ultimately, our aim is to provide you with a gold standard set of metagenomics analysis tools that work together to reveal the key features of microbial communities within and across your sample groups. Finally, by integrating your metagenomic data with your metabolomic and phenotypic data, our platform helps you uncover the functional consequences of those microbial shifts, which offers deeper insights into the complex interactions between hosts and their microbiomes. Thanks so much for your time today, and I hope you feel better equipped to unlock the full potential of your microbiome data with Metabolon's microbiome solution.
Let me just share my screen again here. One moment. Okay, that's working. Excellent. All right, Joshua, thank you very much for that excellent tour of the microbiome analysis tool. For those of you that have done this kind of analysis before, I hope you see a lot of the tooling you already know and love wrapped up into a really collaborative environment. Both you and perhaps some of your less computational colleagues can quickly leverage to bring your creativity to the research challenge. And for any absolute newcomers to metabolomics or microbiome research, I hope you can see a path forward to high impact research in this end-to-end solution. So to recap, the microbiome research solution is now available, combining tailored metabolomics, right-sized metagenomic sequencing services, intuitive bioinformatics, and also comprehensive reports. Together, we really believe that this is an approach that can accelerate microbiome research in a way single omic piecemeal approaches cannot. With that, I'd like to thank everybody, and Josh, thank you very much for the demo as well. I wish everybody a very good day.